If you just do it, it'll turn out okay. Hey everyone, we're back for Talking Time with Caffeine. I just got done watching Dapper Dino watch a Kent video, very ranty and stuff, and now I need some education in my brain. You don't know Tony, but today we have a new guest today.
Is this your first time, Dan?
This is my first time talking on this show. So, hello, thank you for having me. I'm Dan. My channel is Creation Myths, and I'm an evolutionary biologist. So, and my specific training is in the evolution of viruses. So, this is a really interesting time to be somebody who worked on viruses in grad school. It's never good when this many people are interested in what I did in grad school. It's not a good sign.
Yeah. Oh, it's more interested in my subject in tech school. I worked on printing presses in school.
I didn't do any studying on viruses at all. I've never done any of that, but I do a lot of research, as you guys probably know, and this is something I'm fascinated by. So I'm glad you guys had me on.
Awesome.
Tony, I'm getting a little bit of roboting. Is that on my end or yours?
I hear it too.
Okay.
But anyways.
Okay, then it's on my end. I'll fix it. See what I can do.
Sorry.
So let's start with, I guess we start with the basics. What are viruses?
What are viruses? That is a fun question. So I always define viruses as a subcellular, intracellular parasite. So something that is smaller than cells, that doesn't have all the components of cells, but it gets into cells and parasitizes them. It steals their stuff to make more of itself. And then the other kind of side of what a virus is it has to transmit to a new host cell via some stage where you've got a protein coat with the DNA or the RNA on the inside. So those are kind of the two components of being a virus. You got to be less than a cell, get into a cell, take it over, and then you have to transmit to a new cell via what's called an encapsulated stage. If you do those things, you're a virus.
Yeah, I think some people are confused with viral infections and bacterial infections.
Yeah. So a bacterial infection is an actual cell. It has different components, and bacteria they have what's called a plasma membrane. They have a cell wall, and then because they're cells, they kind of can live on their own. They don't need some other entity to like do a bunch of the legwork to make more of them. So what cells that make us sick do, like think about like bacteria that cause like strep throat or something, get strep bacteria in your throat, it'll live on your tissue and damage your tissue, as you know, just that's how it makes its living, whether it's in your throat or, you know, an ear infection will be in your middle ear, it could be in your lungs, it could be anywhere in your body. And the important thing with the bacterial infection is that, at least for now, they are susceptible to antibiotics, which are a extremely diverse group of chemicals that are actually naturally occurring. They're just part of the natural arms race that's out in the environment. And we've taken these chemicals and used them medically to kill the bacteria that are infecting us. Now, viruses, you can't treat with antibiotics. They're not susceptible to all those different things. So you need different approaches when you're dealing with a virus. So you get different antiviral medications, or a kind of a better approach is to use vaccines to prevent the infections in the first place.
Yeah, I speaking of vaccines and viruses, I heard some people they're trying to treat, instead of antibacterials, they're treating bacteria with viruses.
Yes, they are. It's one of the coolest things, and it's something that's going to be like a thing. I don't know when, but we're running out. You've probably heard that we're running out of antibiotics, that the rate of antibiotic discovery is very low. We're not finding new antibiotics as rapidly as we used to.
And the bacteria are becoming immune to them too.
And the bacteria are becoming more immune faster. So they're developing resistance faster and faster, to the point where what used to be the threshold of resistance where we wouldn't use an antibiotic clinically anymore because the dose would be too high and you'd have too many side effects. We've actually lifted that threshold to ensure that we can keep using certain types of antibiotics in certain settings. So the alternative to antibiotics is something called phage therapy, because there are viruses called bacteriophages that infect bacteria. These are phage means eat. So bacteriophages, the word literally means bacteria eater, and they're viruses that infect and kill bacteria. So what you do with this is you take a culture of whatever bacteria it is that's causing an infection. You grow it up in the lab. You allow viruses in the lab to infect that bacteria. So the viruses get really good at infecting that particular type of bacteria. And then you give the person with the infection the viruses, which then kill the bacteria that's causing the problem. It's a really cool approach. It's not approved in the United States yet. I believe, I'm sure someone can give me the specifics on this. As of a couple of years ago, I think there was a phage therapy trial going on. I don't know what the status of that is right now. But if you have the money, you can go to Warsaw or Tbilisi, which is in Georgia, Asia, Georgia, not United States, Georgia, because those are the locations of the former Soviet phage therapy labs. And during the Cold War, the Soviet Union had a robust program of phage therapy. So we actually have, this is a technology that we already have, and it's already used clinically, just not in the United States, but it will be at some point in the near future. We're not talking decades and decades from now. We're talking maybe 1 or 2 decades from now.
And like the antibacterial, if the bacteria become resistant to the virus can just adapt to the bacteria again.
Bingo! The viruses also evolve. So you get this arms race between the bacteria and the virus. So the bacteria will develop resistance, but the viruses are also an evolving population. So they're going to be able to counter that resistance pretty readily. That's what we see in the lab. And go ahead.
As I said, and from what I've been hearing about this virus, that I was afraid of, it's already starting to adapt. So I was hoping maybe it still will be. I was hoping it would be like a one shot and done thing, but maybe it like the flu and we had to take multiple shots.
That may end up being the case. We don't know. What's nice so far is with this. So people have probably heard of this new variant of the coronavirus that I think it maybe first appeared in the UK, and it's now in a couple of different places. There isn't a whole lot of information about it yet. But what we have seen so far is that the vaccines that we have are working just fine, even with the new variant. So that's good. That's lucky. What may end up happening is it may end up in a situation where it's similar to the flu, where you get a yearly kind of thing because it's a slightly different strain that circulates. With like the group of viruses that we're talking about are the coronaviruses. And this is the 7th. So this is SARS coronavirus 2. This is the 7th coronavirus that infects humans. And so 4 of them just circulate and cause the common cold. 2 of them were pandemics. So you had the SARS pandemic back in 2003, 2004, and then MERS, which I think first emerged in 2012 and kind of on and off until 2014. And then it pops back up periodically, but it's not a huge problem. And then so there were 4 kind of common cold ones, no serious illness, 2 pandemic ones that were very dangerous. You're talking like 10 to 20% mortality rates with those. And then this one, which is kind of in between in terms of transmission and being dangerous. What may end up happening, because those normal common cold ones, you get one, and then you're resistant to it for like 2 to 3 years, 1 to 3 years. So it may be the case that with this one, it's kind of a rolling kind of thing where you get a vaccination for it or you get infected by it, and then you're good for 2 or 3 years, and then you need another vaccination or you're once again susceptible. We don't actually know, because the other 2 pandemic ones, the SARS-1 and MERS, we don't have good data on them because those pandemics ended.
Yeah,
so we don't know what the long-term situation is.
I don't remember those 2 that well because I don't remember it wasn't that big on the news or anything. But this one literally shut everything down.
They infected far fewer people. SARS-1 only infected each one, I think, only infected a few thousand people worldwide. It was basically they infected far fewer people, but the case mortality rate or the case fatality rate was somewhere in the neighborhood of 10 or 20%. For I forget which one was which, but they were both extremely dangerous. To put that in perspective, if something has a case fatality rate of 1%, that's incredibly dangerous. So 10% is almost unheard of. That's extremely dangerous virus. Yeah, and there were a lot of precautions. I remember there were a couple of cases in Canada that had everyone kind of freaked out a little bit for the original SARS, but it kind of got tamped down pretty quickly, and it petered out. It didn't become like this worldwide thing that we kind of lost control of, like what happened here.
Yeah, see the vaccines. I heard with chickenpox vaccine now, I missed it by a few years because I didn't like the early.
Me too.
80s. I like in the early 80s, so I ended up having it a little bit later after that, so I just missed the vaccine. I
remember. It's funny, my siblings and I actually straddled that. So I got chickenpox when I was in first grade. My whole first grade class got it. There were out of 27 people in that class, I think like 14 people got it. It just went through everybody. But my two younger siblings got vaccinated against it and they never got chickenpox. So we're just a couple years apart. So we straddled that line there. So now this vaccine is really cool because it's this new type of vaccine. It's this mRNA vaccine, which is super exciting. And this is the kind of thing that I've been telling my classes for years. I talk about the immune system, I talk about vaccines, and I've been saying, okay, we have this type of vaccine. And there's also this fourth one that we don't have any yet. But when we do, it's going to work really well. And hopefully we'll be able to use these soon. And then now for the first time, not for, a great reason, but now for the first time, I'll be able to say, and here's this new kind, and this is the vaccine that you all just got, and that'll be interesting to change up how we do that discussion.
We have a question for you.
Oh, let's see. Question: why are extremely lethal viruses far less common and than less deadly viruses? Great question, because you would think like a virus's job, right? It gets into a cell. It kind of bull in a china shop, it takes over, it breaks the cell, it makes a bunch of copies, and it gets out of the cell and infects more stuff. You'd think the better it is at that, the more dangerous it would be. So the better viruses, the ones that are more successful, would be more dangerous. But you hit a point of diminishing returns because you're a parasite. And if you're a parasite and you kill your host really fast, that doesn't help, right?
Yeah,
that actually hurts you.
You gotta find new hosts.
You got to find a new host real fast. Now, there are some viruses that make that work. Like, think about Ebola, right? I don't know how much either of you or anybody in the audience has read about Ebola virus, but Ebola is really gross. It basically separates all the layers of cells in your body from each other, and then you just bleed like everywhere. And it spreads through fluids like that. So the worse your symptoms get with Ebola, the more it spreads. But with many viruses, that isn't the case. And kind of an example of this, it's overly simplistic, but think about like a common cold versus the flu. Now, a common cold spreads really easily because you get the sniffles, but you can handle it. You don't have to take a day off from work. So you're out and about, you're blowing your nose, you're washing your hands, hopefully, but probably not perfectly every time you blow your nose, right? You're gonna, touch doorknobs, you're gonna shake hands, maybe. You're going to be in the same room as people. So that cold virus is going to spread around. But if you have the flu, I don't know if anyone's had the flu recently, but having the flu is awful. Everything hurts. You don't want to get out of bed for a week. You're just not encountering that many people. Now the flu, because the symptoms are far worse, it actually inhibits its ability to find additional hosts. So that's kind of an idea of that trade-off. That trade-off does not always hold, but it does in a lot of cases. And that's what's going to, so you have kind of this trade-off between how dangerous are you versus how efficiently can you spread through a population. That's sometimes the case.
Now, I might be wrong about this, history, I might have heard this wrong, but if and the history might have been wrong too, obviously, but the first vaccine that I know about was the Calapox Smallpox vaccine.
Yeah. It was Edward Jenner in the 1800s, And it's a great story. Can I tell the story? Is it okay if I nerd out and tell the story? It's so cool. So this was, and that's actually where vaccinia comes from. It comes from vaccinia is the name of the organism for cowpox. I think it's for cowpox, and the word vaccine comes from that, because the cowpox virus was the first vaccine. And what happened was, it was during the Napoleonic Wars. Somebody realized that for whatever reason, cavalry soldiers didn't get sick when there was a smallpox epidemic in the army during the Napoleonic Wars, and technically it's the Wars of the French Revolution, and so whatever, but late 17, early 1800s. And what happened was they eventually realized, and the other group of people that didn't get sick from smallpox was milkmaids. What people realized was that they were actually infected by cowpox, which could infect horses and spread from horses. And they would, if you got cowpox, that would confer resistance to smallpox. They were similar enough that the immunity from a cowpox infection made you less likely to get severely ill from a smallpox infection. So they started inoculating people with cowpox on purpose. They'd give you cowpox on purpose, and that means you wouldn't get sick from smallpox. And that is, I'm not exaggerating to say that single discovery, probably more than anything except sanitation, has saved more lives than any other invention, probably with the exception of sanitation. It's just like, that is one of the most incredible, important discoveries, not just of the last 200 years, it was a little over 200 years ago, but just ever in the history of humanity, in terms of improving the general safety of being a human.
And is that pretty much how vaccines work even today? You give yourself a weaker version of the virus, so your body can get used to, your body knows what it can get used to attacking it, and then when
some of them,
sees the real thing, it's like, oh, I know that thing, kick its butt.
I know that thing. Exactly. So some vaccines still work that way. So there's a bunch of different types of vaccines. You've got your inactivated vaccines, which is where you take the virus or the bacteria, because you can vaccinate against viruses, bacteria. You could vaccinate against big parasites, eukaryotic parasites. So there's now a vaccine against malaria. There's a malaria vaccine now, which is a eukaryotic parasite. So the idea with these attenuated vaccines is where you take the thing that makes you sick, you grow it in a different environment. And like for polio, for example, the famous for most of the 20th century and into the 21st, the polio vaccine was an attenuated vaccine. It was the actual polio virus. It was just weakened because it was grown on monkey cells. And when it mutated and got better at growing in monkey cells, it got worse at infecting humans. So you could get infected with this weakened polio virus, and before the polio virus mutated again inside you and got dangerous, you had built up the immunity to it. So that's one approach. Another approach is called an inactivated vaccine. You take the pathogen and you kill it with like chemicals or heat or whatever. You just boil the thing. Okay, you've got your intact virus or your intact cells, but it's dead. And you inject those, and your immune system still sees them and still, freaks out a little bit and recognizes them. So like the flu vaccine every year that you get injected is an inactivated vaccine. There's whole flu virus in there, but they're dead. And then the third type, the second newest type, is called a subunit vaccine, where instead of the whole virus, it's just the piece that your body recognizes, whether, the protein that sticks out on the surface maybe, that your immune system sees and makes antibodies to. You just inject a whole bunch of that. And then when you see the real thing, you go, aha, I've seen this protein before. You don't care what the protein's attached to. You just see the one thing, and you can attack that. And then the newest one, of course, are these RNA vaccines, where you bring in the RNA, which is the nucleic acid that your cells use to make proteins, and you bring in the RNA to make those surface proteins for the virus. So you make your own, and then you learn what to target. And the advantages of that last one are that you can get a much higher dose of the protein, because each little piece of mRNA is going to lead to a lot more proteins than you could fit into a single shot. So these RNA vaccines, awesome. Like this is potentially groundbreaking. Now, if we're lucky, we can now vaccinate against things that we previously couldn't, because we couldn't generate a vaccine that caused a robust enough response. with the RNA vaccines, we might be able to do that now. So this may open up a lot of doors into new preventative measures, which is really exciting.
Yeah, and like each virus can with not vaccine, they attack different types of cells.
Usually, yeah, it depends on the virus, but not always. So, like, influenza is really picky, right? Influenza attacks just your lungs. It doesn't attack anything else. It doesn't replicate, like, in your nervous system or your intestines. It just does your lungs. But the funny thing is, in different species, influenza viruses do different things. So, like, everyone's probably heard of, like, avian flu, and we're always worried if, like, an avian flu is going to jump into humans. Well, avian flu doesn't affect bird lungs. It affects their intestines. But when those viruses mutate just a little bit and they jump into humans, now it's a respiratory virus.
Yeah.
But they tend to be specific. Now, that's different for other viruses. Ebola doesn't seem to care that much. That just goes wherever it wants and it infects whatever it wants.
Like the AIDS HIV virus, they attack the white blood cells that attack other viruses, which makes them dangerous.
Devious. Yeah, right. Because it makes if you're affected by that, then it makes you susceptible to lots of secondary infections, because the virus basically does a the way I always think about it, it's that scene at the end. I don't know, this is an old reference. I'm sorry, everybody. But in the original Men in Black, where Will Smith, it was Tommy Lee Jones gets eaten, right? And then he blows up the alien from the inside. Sorry, spoiler alert. That's basically what the HIV virus does. It basically says, it doesn't try to hide from the white blood cells. It says, hey, I'm over here. And then the white blood cells engulf it. And that's how it gets in. And then it takes over once it's inside. Devious.
And then you could die from a common cold even, because your cells aren't there to attack the other ones.
Yeah. Yeah, the dangerous thing there are infections that aren't dangerous to non-immunocompromised bodies. So, I mean, you're talking, a yeast infection, Candida albicans, or a type of virus called cytomegalovirus, which everyone's infected with cytomegaloviruses. By some estimates, our immune system spends like 10% of its energy dealing with cytomegaloviruses, but they're easy to deal with. They don't make you sick most of the time. But if you're immunocompromised, they can cause all sorts of problems. There's types of pneumonia that you can only get if you're immunocompromised generally. That was actually how the AIDS pandemic was found. It was a rare type of pneumonia that was documented in a couple of different places. And it was literally one person put two and two together and wrote it up in the CDC. It's called the MMWR, the Morbidity and Mortality Weekly Report. And it was in like 1981, this one person, she was just like, you know what, this is weird with this pneumonia over there, and we've never seen this before. And that's considered the beginning of the AIDS pandemic, because it was the first time anyone had said, like, something weird is happening here.
Oh, yeah. But vaccines and anti-vaxxers, are they a new thing, or they've been around for a long time? There's now internet.
Forever. There are political cartoons from Jenner with anti-vax propaganda. They've been around as long as vaccines have been around, as long as the concept of vaccination has been around. You can find it's called, I just Googled it, it's called the Cowpox. It's called the Cowpox. And it's a old-timey political cartoon from like 1800 or so. And it's Jenner injecting people with cowpox, and then those people are subsequently turning into cows. So, yeah.
Yeah.
Anti-vax propaganda has existed as literally as long as the concept of vaccination has existed. The internet's not helping, but it's certainly not new.
Yeah. Yeah. My favorite one is the vaccine gives you autism. I'm like, okay, I'll take, if it's true, I'll take the autism.
I mean, that goes back to literally, there's one study. It was 1998, I want to say. It was Andrew Wakefield. And this guy's a British doctor at the time, although he not anymore, because he can't do what he did. So what he did is he was coming up with, so he was targeting specifically the trivalent, the MMR vaccine, the measles, mumps, rubella. And not by coincidence, he was developing a single version of one of them. I forget which one, measles, I think, but don't quote me on that. And basically a competing treatment or competing vaccine kind of course to the MMR course. And he literally paid like the participants, he like paid parents to have their kids produce like blood samples for his work. It's not even enough to say that it was fraudulent. Like, he literally just made stuff up. It's insane how like blatantly just lying he was. He literally just made stuff up for his famous paper that was subsequently retracted, and he lost his medical license and yada yada yada, but the damage was done. You can't unring that bell once you publish such a study. I mean, my goodness, everything about that paper is just wrong. And it goes back to Wakefield basically having a financial interest in the MMR shot having a problem so that his competing product would be used instead. And to say it was fraud is an understatement. But apparently, even so, from what I hear, even then he still believed in vaccines because he wanted his vaccine. Still believed in vaccines. He just wanted you to use his vaccine, not this dangerous one over here. Yeah. Mm-hmm.
But then other people took it off in a whole direction.
Now we're just gonna go. We're gonna take that and turn it up to 11. Yep, that's it.
Yeah. Like I said,
here we are. And
yeah, I have a friend whose mom has polio because he was born before the polio vaccine. So my friend is really for vaccines because he's seen her mom suffer through it.
Yeah. Oh yeah. It's a lot. I think a lot of it is generational because like people my age, your age, we're pretty close in age. We didn't grow up with people getting measles. We didn't grow up with people getting mumps or rubella. Like that stuff was, polio. That stuff was largely gone. So we didn't have friends in elementary school that got polio and died. We didn't have any, but we don't know anybody as kids. Didn't know anyone as kids that got measles and got really sick. I didn't know anyone that got measles as kids, right? Because everybody was vaccinated. So we get this kind of false sense of security, and you get anti-vax people that are going, why do we need to do this anyway? Look at how few cases of measles there have been in the last 20 years or whatever. It's like, well, the reason for that is because everybody is vaccinated. If you're standing in a rainstorm, but you have an umbrella, you don't go, oh, I'm perfectly dry. I guess I can put the umbrella away. Like the vaccines are what is making it safe. The fact that it's safe doesn't mean we can stop vaccinating. It just, it makes my head hurt.
Yeah,
it makes my head hurt.
I have a few seasons ago. I had a friend who literally can't get vaccines because her body can't handle it. So she relies on other people doing like herd immunity or whatever.
Exactly. So you have to make sure that enough people are vaccinated so that the pathogen can't spread. So you can cover the people that are immunocompromised, that have an allergic reaction, that are on chemo, like all these different groups of people that can't get vaccinated. It's on everybody else to vaccinate so we keep them safe. Yep.
Yeah. Oh, so how come, I guess it probably is evolution, but like some viruses like the measles, mumps, polio and stuff, chickenpox are one and done, but other ones like the flu are seasonal?
Yeah, it's a great question. So some viruses, it has to do with what the vaccine targets. And certain viral proteins are going to evolve really fast, and other ones are going to evolve much more slowly. So like the influenza, for example, the targets for that, because of the structure of the virus, the targets for that vaccine are the ones on the outside that interact with our cells, not just the cells they infect, but our immune system cells that are like, aha, this thing. So the virus, we actually, by the virus circulating, by us vaccinating, we're imposing a selective pressure on that virus for the rarer variants. So if you have a variant that's really common one year and you do a really good job immunizing everybody against that variant, well, that variant is going to be really rare the next year, right? And a rare variant is going to become more common in its place. So the vaccine that you had last year, that immunity works. You'll still make antibodies to that virus. but it's a slightly different strain of that virus that's going to be circulating. So your antibodies from last year aren't going to work. So you need a new flu shot. And that's kind of the challenge with influenza. And we talk every year about, like, is it going to match? Is it going to work? Some years it doesn't work. And it's because by like March, April, May every year, you have to look at what the trends are for the different strains in the influenza population and make a projection about what they're going to look like in September and October to have that vaccine ready by September for the next flu season. So you have a really quick turnaround, and you're basically taking a couple of years worth of evolutionary history and then trying to project forward for the next 6 to 8 months, which is an extremely challenging problem.
But the older ones, like the measles and stuff, they don't evolve that fast, or didn't evolve.
They evolve, yeah. The target that we use for them, and I don't know what the targets are for many of these, all of them, but the targets that we use are stable. For whatever reason, some viruses, they don't change things very much. Maybe it has to do with if there's a mutation to that protein, it doesn't work. If it's part of the coat or something, maybe any mutations that would allow it to evade your immune system based on the vaccine, any mutations might inhibit it structurally, so the virus wouldn't work as well. I don't know, and I'd expect it to be different for as many groups of viruses as there are. But it's just the case that some viruses evolve slow enough that we can vaccinate once during childhood, and you're good for decades. And then what we're finding is, in some cases, it seems like decades later, not 5 years later, but maybe 20, 30, 40, 50 years later, maybe you need a booster to just make sure you're good.
Is that like with the, like, was it with the shingle shot in your 50s, whatever?
Yeah, that's a weird one, because that's actually the same. That's such a weird one. That virus is weird because that is the same virus as causes chickenpox. It's the same virus, but what it does is you get chickenpox, and that's like the acute infection, and it infects your skin, and you get angry, red, itchy spots all over your body, and then it goes away. Your immune system defeats it, and it's done, and that's not going to happen again. But those viruses that hang out in the cell bodies of your neurons, specifically neurons in your spine, where if you could picture this, you got your spinal cord, right? And you've got neurons that have the cell bodies in your spinal cord, but then the axons, they radiate to either side. So they go out, to your ribs, your arms. Some of these axons can be like several feet long. And it's one cell. It's just this long, spindly, basically a wire where it sends signals. And the viruses will go into this kind of dormant state. The intact viruses, and they'll just kind of go into stealth mode, and they'll hang out inside the neurons for decades. And then later on, they'll just be like, I'm here now. And they'll ride that axon out to the surface of your body. And that's why you get like weird little patches on the periphery for shingles. It's whatever neuron happened to be infected. It's where does it end? That's where you get the rash on the surface. It's such a weird virus. I don't know of any other viruses that do that exact thing. But then basically you can, once you've gotten either chickenpox or chickenpox immunization, you can then have a secondary immunization to prevent that other thing from happening. It's so weird. It's so weird.
Really quick question for you.
Question?
Yeah.
Was there a question in the chat? Oh.
Yeah, right there.
Oh, yeah, 100%. So what I would say is it's not that they evolve like to be vaccine resistant, because they're not like trying, but what'll happen is vaccines can actually impose a selective pressure. So if you have a population of viruses, right, and 90% of them are susceptible to the vaccine, meaning if that population gets into somebody who's been vaccinated, 90% of those viruses, say, are now going to be targeted by the immune system. Well, which viruses are going to be the ones that survive and replicate and spread to a new host? It's going to be the small fraction of the population that was not susceptible. So the vaccines, it's similar to like antibiotics, where by using it, you're imposing a selective pressure and potentially promoting the variants of the virus that are not susceptible.
And the ones that don't, the ones that survive, that like people, they tell you to take all your medicine. If the virus, if the bacteria or the whatever survives, it can mutate
exactly,
and not to be able to like, oh, we survived this,
right? So
anything.
Yeah, so with viruses, you can't, a vaccine is an on-off switch, right? Either you get the vaccine and you get the response or not. With bacteria, it's a little bit different because it's You could feel better 3 days into a 5-day course of antibiotics. The problem is you haven't killed all those bacteria, and the ones that are still alive are the ones that survived 3 days of antibiotics. You want to kill those. You want to make sure those are dead, because when they come back, if they've already survived 3 days worth of antibiotics, you're not going to feel better after 3 days on the next time if they come back. So that's why you got to do. That's why you got to do the whole thing.
So, let's see.
Oh, I see a lot of good questions in the chat.
Okay.
These are good. These are fun questions.
So you could want to click one and answer it.
Yeah, here. How about I just see Scott Duke saying, if viruses aren't life, how close do they come to being life? What is the scientifically defensible definition for life? I love this question because so biologists love to do this thing where we put stuff in boxes, right? We say, this is a species. Everything goes in that box. This is a different species. Everything goes in this box. These things are alive. These things are not alive. That's dumb. We shouldn't do that. There's a continuum. And so you can look at cells, right? Cellular life. Generally, if you're alive, like by the high school definition of living, you have to have cells. But there are cells that are really tiny. They're parasitic. They're maybe missing parts of their genome that like do important things. So they have to live like attached to a bigger cell.
Yeah, it was good that. I was wondering about this. Like if maybe I don't know how it really works, but if RNA viruses were the first life forms, and then some of them split off to become the first cells with DNA, and the other ones became the viruses with the RNA.
I have a weird theory about this, and I have no support for this idea other than it makes sense based on what we see in the world right now. But what we see is overlap, right? So in all the activity of like what cells do in terms of their size, their biochemical complexity, all the stuff you see, a big range. And then viruses typically do less than all those things, but they overlap the cells. So if you got your cells, I'm trying to keep this framed here. So you get your cells like this. Your viruses are maybe like this. Generally smaller, less complex, but there's overlap in terms of the size of the genome and the number of genes. There's overlap between the biggest, most complex viruses and the smallest, simplest cells. And then you have other entities, things like plasmids and viroids, transposable elements that are smaller and simpler and less complex than viruses, but there's still overlap in there. So what I think happened early is you had a bunch of just reactions going on, and different parts of those reactions got kind of contained within compartments. One type of compartment became cells. Different types of compartments became the different types of viruses. And one thing we know about viruses is that unlike cells, they don't share a common ancestor. Like all cellular life, from humans all the way down to the smallest, simplest bacteria, we all share a common cellular ancestor. But that's not the case for viruses. All these different groups of viruses have independent origins. So there are multiple independent origins for viruses by multiple different methods. There's one group of viruses that are really big. It's possible that they're like degenerate cells, basically parasitic cells that started transmitting via like a viral-like state, right? There's another group of viruses. It's a group that infects plants that it was a plasmid. It was a completely different thing. There's this little circular piece of DNA that infects bacteria, and it stole a capsid gene from a different group of viruses. So you could take this group and divide its genome in half, basically. Half the genome matches up with this one group of plasmids. The other half matches up with this completely different group of viruses. And it's this weird chimeric genome, completely new group of viruses. Like, they're super weird, and there's no one origin. So trying to draw a line between viruses and cells in terms of living, non-living, I don't think it's a useful approach because there's so much overlap and interaction between all the different components. Plot twist. It's the Andromeda strain. It's actually the Andromeda strain, everybody. They came from outer space. That's it. It's over. I actually see one question here that's kind of related to that. Is it possible for a virus to get infected by another virus? Yeah. There's a group of viruses called virophages that basically they can infect a host cell, but when they infect on their own, they can't replicate. But there's a bigger virus that infects that host cell and takes over. And then when the virophage infects, it takes over the bigger virus's machinery and converts the big virus. Instead of making more of the big virus, it just makes a lot of the little virus instead. It's called a virophage, which is actually, some people will consider that evidence that those giant viruses started off as cells. And then the virophages infected the cells and stuck with them as they kind of shrunk down into a group of large viruses.
Yeah. On the same, back up a bit. On the same, I remember something that happened earlier last year. On the same phase as anti-vaxxers, the anti-maskers that attacked this year, movement attacked. My favorite thing was, like people say they couldn't breathe through the vaccine. They couldn't breathe through the mask. And they said, "Well, if you can breathe through the mask," people would say, "If you can breathe through the mask, then you can get the virus through the mask." And I'm like no. It's like viruses may be small, but you know what's even smaller than viruses? Molecules, like carbon dioxide molecules.
Yeah. The whole I can't believe that was on it. Like, I am astonished that was a real argument that people were making, but people were actually saying that the masks, if they're able to block the viruses, then they will also block oxygen. And for anyone who doesn't know, oxygen that we breathe is 2 oxygen atoms stuck together. It's O2. And a virus is like millions of times bigger than that. You
don't mix up a viruses, atoms and molecules, everything else.
A lot of them. And the coronaviruses aren't even particularly small as far as viruses go. They're not huge, but they're also not tiny. They're pretty complex. I mean, they're enveloped viruses, so outside of their protein coat, they have a membrane. They're pretty large. Not, I won't say they're large, but they're not tiny in terms of viruses. There are smaller viruses that are much harder to filter out of the air than what we're dealing with right now. There's a reason medical masks are designed the way they are. Like, there's a reason they're designed specifically that way, because you use them in a medical setting to prevent things like viruses from transmitting.
Yeah,
what they're designed to do.
My other argument was that I can't wear a mask for 10, 20 minutes to go to a store to get something. And lots of doctors have to wear a mask for 6, 8 hours in surgery.
Yeah, 6, 8, 12 hours. I mean, no. Wear a mask. Wear a mask, everybody. Even as the vaccines are rolling out, wear a mask just to be safe. Because, we don't know, there are no documented cases of anyone that has gotten vaccinated subsequently transmitting the virus. There are only a handful of cases of people getting vaccinated and then actually getting the virus. Of those, there have been no documented cases of transmission from someone that was vaccinated. That doesn't mean it can't happen. So until we get the all clear, even if you're vaccinated, when you go to the supermarket, you're going out in public, whatever, still wear the mask.
Yeah, that's an American thing, or is that in the other countries of the world too?
I mean, I think a lot of it is probably the American, individualist thing. I think a lot of it is the American anti-science, the reflexive anti-science thing. I also think just in general with the masks, there is a healthy dose of, I don't know if racism is the right word, but kind of, I don't know if xenophobia is too strong, but basically looking at it as primarily a thing that people in East Asian countries would do, and not particularly effective, kind of quaint, but they do it and it makes them feel better, and that's fine. And it turns out, rest of the world, that the public health experts in those countries actually do know what they're talking about. And they actually are effective at preventing the transmission of not just SARS coronavirus 2, but also things like influenza and common colds. Because one thing you'll see this year is that we're actually, so far, fingers crossed, having a mild flu season, because all of this behavior like masking is also limiting the spread of influenza. And that's really useful. And even once we're done with this pandemic, that's something we should keep. We should, it should just be a general thing that if you're feeling, if you got a cold or you think you maybe have the flu, and you have to be around people, wear a mask. Just common decency, wear a mask.
Yeah. So I remember it's going to end when it gets warmer, or it's just going to end when it gets hot, or it's going to get in the fall when the flu.
I'm old enough. I'm old enough to remember when there were 15 cases, and in a few days it was going to go to 0. I'm old enough to remember that. So the thing that surprised me about this whole thing, actually, I don't know how political you get typically, but I
I usually don't, but like a few weeks ago, me and Tony and Eric were talking about the election fallout and how that happened. So if it's that big of an event, then I might talk about it.
I mean, the thing is, like, I had conversations, like, my relatives, I remember like it was yesterday, last February, my relatives would be like, what's going on with this virus? Should we be worried? And my response was like, look, we deal with this kind of thing pretty regularly. You know, we had the SARS pandemic in 2003. There was the H1N1 in 2009. There was MERS in 2012. There was a 2014 Ebola outbreak. Like, there is a pandemic playbook. There is a pandemic playbook.
Yeah, but back then we had people that actually knew how to deal with it. And
I figured, silly me, that we were just going to follow the playbook. Because, like, there is a way to deal with pandemics. And, like, it's not a secret. Like, look at places like New Zealand, Vietnam, South Korea. Like, there's a playbook. Oh, there's a pandemic. Okay, let's do all these steps that we know is how to solve that. Like, there is a playbook that you follow, and if you follow all the steps well, you don't have a bad pandemic. And we just, not only did we not follow the playbook, not only did we not read the playbook, we set the playbook on fire and threw it away. Like, we were just like, we're going to do the opposite of what you should do in this situation. And that is what surprised me the most of any of this, that we just completely said, you know what? We know how to deal with this, and we're just going to do the opposite of that. And like, I am still shocked that we just collectively made that decision.
Yeah, I heard Clorox kills viruses. Let's get some Clorox.
Man, it's I will never not be shocked by that, because literally, like, and not even like, okay, you don't want to have it. Nobody wants to have a pandemic on their watch, but like, you know, like, there's an alternate history where presidents could run for any number of terms, where Barack Obama just cruised to his 4th election on the strength of his pandemic response, because he shut that thing down and we had minimal disrupt. Like, there is an alternate universe where that happened.
Yeah, that's probably why we didn't hear of the 2014 pandemic, whatever, the 2008 pandemic wasn't that big of a deal. I mean, probably because we took care of it.
We had a playbook. We had a lot, like the 2009. I was in grad school during the 2009 swine flu pandemic. It was fascinating because I got to watch it in real time. As like, and I like, my stuff was about viral evolution. And some of my colleagues, like, they actually tracked disease outbreaks. They didn't track human disease outbreaks. They tracked like disease, plant pathogens. So things like cassava mosaic viruses and tomato yellow leaf curl, stuff like that. And you know, we're watching this in real time as they're tracking the 2009 strain and the H1N1. And it was just, you know, you had massive surveying. Anytime there was a potential flu case, it was, what is it? Where are they? Isolate, contact trace. Like, it was on the ball. And like, it's hard. You know, it takes effort and experts and money to do. And we just decided not to do it. And here we are. And yeah, I see, you know, like Australia did well, but a bunch of businesses went out of business. I would put money on it that it's more than 7% here, because like, having to shut down and not have customers that's a big problem. But you know, it's a worse problem. Having a pandemic where 500,000 people die is a worse problem for business. Like, man,
you know, my favorite response was, like this virus is only killing 1% of the population. I'm like, that's still 1% of the population. That's still a lot of people.
That's,
you know,
great. Let's get 100 random people and see who volunteers. Like, come on.
Be like 1% of 7 billion is like what? Still 700, like 70,000 people, 700,000 people. It's still a lot of people that are dying.
It's a lot. It's a lot. Now, for what it's worth, what we're finding out, I think is probably the case, is that the case fatality rate for this virus is lower than we originally thought because in the spring, we just didn't have the testing capacity. So if you look at the graphs, right, they look like this, right? They got a bump in the spring, and then the summer's not bad, and then there's that summer bump, and now we're in the big winter bump, right? But that spring bump from like March and through April, that's probably about 1/10 of what it should be.
Yeah.
So, we just missed so many of the cases.
But Dan, if we stop testing people, we'll stop getting so many results.
That's the magic, right? That's how it works. Man.
Yeah, and what was that? I don't hear that much anymore, maybe a little bit, but what was that thing that when they first started about the medical bump thing? So they keep it under the loop or something curve.
It's called flatten the curve, right? So the point was, if you could picture it like this, you have some kind of capacity of like ICU beds, ventilators, whatever you need for treatment, you have a capacity for that. And if you have more people sick that need that stuff at any given time, if you exceed that capacity, then that excess is much more likely to die because they're not getting the treatment they need. So the original idea with the flatten the curve thing was, if you look at those early projections, you're going to blow right past your capacity, and a lot of people will die unnecessarily. So what you want to do is take that really tall, steep curve and make it wider and flatter. So even if the same number of people get sick, even if the area under the curve stays the same, at no point do you cross that threshold of your capacity of your medical system. So even if everyone is getting sick, the same number of people ultimately, it's happening over a longer period of time. So you don't exceed your capacity and fewer people die. The other side of that is we learn how to treat it better. We're really good at treating coronavirus right now. We're light years ahead of where we were in March, right? We're so much better at treating it now. So by drawing those cases into the future, a case that we may have treated incorrectly in, say, April, right, that maybe would have died, we'll treat it correctly in August and will survive. Right, so that's the other side of it. By drawing it into the future, we're going to be better at this in the future than we are in the present. So the more you can push it, the better.
It's funny. In February, we pretty much rebuilt our whole lobby at McDonald's. Like we were closed for a month. The lobby was rebuilt. We opened up for a week, and then shutdown happened.
So
we spent all that money. I'm like, we can spend that money rebuilding the kitchen, have our equipment's falling apart. We had to get the lobby fixed up for no one to use.
And eventually it'll get there. And like right now, we're now I think I forget if it was today or yesterday. It was probably, we don't have the numbers from today, so it had to have been yesterday. But I read that there were 1.6 million vaccination shots administered. Like, keep that number going. We got another vaccine coming on the scene soon. Two, actually. AstraZeneca and J&J are going to be there soon. I forget which one, but one of them is only one shot instead of two. Plus, it was just reported, I think today or yesterday, that we're going to have enough doses to have 300 million people vaccinated by the end of the summer, which that's it. Like, the problem with that is you're going to have kids who are too young because we haven't completed the trials. The way the trials are working is they're stepping downwards in age. So you get, at first it's 18 and above, then it opens up to 16 and 14. I don't know where they are now.
Like the first, either the older people, and then maybe the people that are like that have pre-existing conditions, maybe.
Right. But then the other problem, getting 300 million people vaccinated, is what we're calling vaccine hesitancy, which is people don't want to get vaccinated, which seems not rational to me.
Because the anti-vaxxers got in their brain, or they are anti-vaxxers.
I mean, it's not even like the militant anti-vaxxers. It's just like the occasionally good faith, poorly reported stuff that you see. Part of the problem is it's a new type of vaccine. People are worried about, like, well, what does this RNA vaccine do? People are worried that it like changes your DNA. But the thing to remember, RNA, and in particular it's called mRNA, is the specific molecule, it's super unstable. Once it gets into the tricky part with these vaccines is getting them into your cells. You have to have like a little lipid particle to do that. Once it gets into your cells, that mRNA lasts about 5 minutes. And in those 5 minutes, you make a ton of those viral proteins that you want your immune system to recognize. And then at that point, it's basically a subunit vaccine, just with a higher dose. And so it's just a little part of the virus that's now inside you that you've made. And your immune system sees it and goes, that's not me. I need to be ready to kill that thing. So now your immune system can really ramp up, and it's a much larger dose, because think about like a protein that your immune system recognizes. You inject a certain number of those proteins, and that's the dose. But if you inject the same number of mRNAs, you make a bunch of the proteins from each mRNA, so you get a much higher dose, a stronger response. Everybody's good. And yeah, it's the mark of the beast. Yeah, it's the that's the technology for the mRNA vaccines is just fantastic. And it's, like I said earlier, because you get a more robust response. One, you feel kind of like poop after your second shot. I was actually in the Pfizer trial, and they just unblinded us. So I actually got the real vaccine in the Pfizer trial back in October, November. And I knew I'd gotten the real thing, because the day after my second shot, starting like 12 hours after my second shot, I had a mild fever, chills, body aches. Like, it felt like I had the flu a little bit. It lasted about 24 hours, but by the next day, by the next evening afterwards, I was fine. It was one day of unpleasant, and I was happy about that, because that meant, one, I got the real thing, and two, it was working. And that was really cool.
That reminds me, I've heard this a few times about anti-vaxxers, that they say they're saying we should have a placebo vaccine for people for trials and stuff.
So we do. This was a full placebo-controlled trial. The placebo was a saline injection. Okay. Now, what they'll sometimes do, because what they want to see is that known vaccine versus new vaccine, what they'll do is, as the placebo, they'll give you a different vaccine, kind of a well-established vaccine. I forget which one they use, but sometimes they use, it's instead of a true placebo, which is just a saline injection, it's a vaccine placebo, where it's a different, well-studied, nothing out of the ordinary vaccine. And then they compare I just got vaccinated with this known thing versus I just got vaccinated with this unknown thing. Right? That's the other way of doing it. All of the trials for this new vaccine were true placebo-controlled trials, with the exception of one, and I forget which one, but one of them did use the kind of vaccine placebo, but the other, like 4 or 5, used true saline placebos. I don't know where they get that from. Like, no, it's placebo-controlled. Like,
what is the point of what do placebos do? What's the point of that?
Nothing. That's the point. It's the point that it has no physiological effect. So you can say if 5% of the experimental group report a certain set of side effects, but 1% of the placebo group report that same set of side effects, then that tells you that of that 5%, not all of them are due to the vaccine. Because there's something else in there, the injection, the volume of liquid, something else about that thing was causing some of those side effects.
Or it could be or it could have been nothing at all. It could have been something totally random, the vaccine at all. If both parties are getting the same thing, and like,
Yeah, and this is important for other treatments, because you get a treatment, right? You get a new drug to treat whatever condition. Well, if you get a placebo, if with that new drug, say you get 10% of people see an improvement in whatever condition, but then you get the placebo and 8% of people see improvement in that condition, well, is that drug really doing anything? Not really. For whatever reason, 8% of people apparently are going to improve one way or the other. So, like, is that drug that improves in 10%, like, worth it? Who knows? That's the importance of your placebo control, is that you need to see if the effect that you're observing is actually from the thing that you expect it to be from, or if it's just happenstance, a side effect, whatever, of the process that you're doing, or maybe it just would have happened anyway.
All right. So, anybody in chat have questions for Dan here?
Questions, please. This is super fun. talk about
Anybody
vaccines,
on
viruses, and stuff all
anybody
night.
here on YouTube, Twitch, or Facebook? Come on down. You're your next contestant. like this.
I have YouTube open, but I don't have the other ones.
Well, you can check this on this comments on the side.
I had was a president who worked at science. I had not my congressman, but a nearby congressman was a physicist, and I think he had bumper stickers that said "My congressman is a rocket scientist" at one point, which I like that a lot.
What's your favorite TV show?
What's your favorite TV show, man? I don't watch a lot of TV. I really don't watch a lot of TV. I will say, if anyone has not watched The Wire is required.
Yeah,
watching.
I haven't watched. It's been years since I've actually watched TV anymore. Since the most stuff's on Hulu or Netflix.
I will say there's an ongoing season of BattleBots, and what can I tell you? I'm in. I am in for BattleBots.
But I would still, yeah. But I still have to say my favorite show, still, Boy Meets World from the 90s. Still my favorite.
Classic. In terms of stuff that's on now, I'm behind, but I want to catch up on The Expanse, and I still haven't finished. I still have a season of Peaky Blinders to do. So those two I gotta get caught up on. Oh, how did HIV spread so quickly? Yeah, because you would think being not exclusively, but primarily sexually transmitted, that HIV would be tough to spread. That's true. The thing with HIV that's so tough is that it has a really long incubation period, depending on the strain of HIV, 6 months to 10 years before you get sick from it. So if you are infected,
you don't know you have. You don't know.
You get an acute illness, and then it kind of goes away, but then it builds back up, and then you can have anywhere from a few months to almost a decade, where you don't really have symptoms, but you're actually able to spread the virus. So if you look back at the history of the HIV pandemic, you see it really explode in the early 80s across 3 or 4 continents simultaneously, boom, boom, boom. Where did all this come from? And one of the initial things that was confusing is because everyone thought it had to be different viruses, because you can't have an epidemic of the same virus simultaneously on 3 continents. It has to start somewhere and it has to move. But with HIV, it crossed into humans probably around 1930, and then it spread at very low levels under the radar for decades. And it didn't start really spreading a lot until the 60s and really in the 70s. But then all those infections that happened in the 70s, you don't start seeing them until the early 80s. And that's when it exploded.
And it's not just a sexual, probably a sexual disease. Yeah, it wasn't just a sexual disease either. Like the famous kid from the 80s or 90s, that Ryan guy didn't have by having sex.
Exactly. Yep. Blood transfusions. Yep. So there were blood transfusions. There was a famous case of where a dentist infected his patients because it was a cut on his fingers in the mouth. That was a very famous case. It was actually the first use of DNA in a courtroom, was determining whether the dentist did actually infect the patients, which is an interesting case study. It's an interesting. If anybody is interested in that, there are 2 books I cannot recommend enough. One is called The Coming Plague by Laurie Garrett, which it's not just about HIV, it's about emerging diseases. So there's chapters on HIV is like a novella in there. It's like a 200-page chapter. But then there's Ebola, there's bubonic plague. I learned from that book that every decade or so, there's a bubonic plague outbreak in the southwestern United States. Just no big deal. It's just something that happens every 10 years or so. There's a chapter on influenza, smallpox, all kinds of stuff. Great book that's called The Coming Plague by Laurie Garrett. And then specifically for HIV, it's And the Band Played On by Randy Schultz. This book is incredible. It's much more of a kind of it's not just about the science. It is about the medical side and the science and the epidemiology, but it's also about the social and political aspect of it. And for that reason, it is absolutely infuriating. And the band played on, but it is a just fascinating read about the kind of the first decade or so history of the AIDS pandemic. It's an absolutely incredible read. Cannot recommend it enough. Oh, favorite virus? I really like the big weird viruses like Mimi virus and the aptly named Pandora virus, which, true story, it's the biggest known virus, and we isolated it from melting Siberian permafrost. And then we said, I wonder if this thing still works, and brought it in a lab and tested it against a bunch of cells, and turns out it still works, even though it had been frozen for tens of thousands of years. But there's also a place in my heart for the virus that I worked on in grad school. It's a teeny little weird single-stranded DNA virus called Phi-X174. And I dig that virus.
Oh, that reminds me, Anthony, that people are saying that this virus was created in a laboratory somewhere.
Oh, you mean the SARS coronavirus too?
Yeah.
No, because we have the genomes. We can look at the strains and we can see where it came from. And it very clearly did not come from the lab in China. It came from bats. You can do a phylogeny of this virus, and it falls out in a clade where it's just bat coronavirus, bat coronavirus, bat coronavirus, bat coronavirus, SARS coronavirus 2, bat coronavirus, bat coronavirus, like it's from bats. We figured that one out. I have not read that book, The Chimp and the River, but I'm going to read that book. Hot spring. I'm not familiar with that hypothesis. I don't know. Yeah, I don't know. Oh, it's recent. That's a real recent one. Yeah, I don't know that one, but I'll have to look into it now. Thank you.
Yeah. So that's it. You read this book? You said you haven't read this book. I am not familiar with that book, but I just wrote it down. I'm going to read it because yes, please. Yeah, it's fascinating. There's a really good Radiolab, actually. It's an old Radiolab episode. It's probably like 8 or 7 years old at this point. But yeah, it's a really good episode on the origin of HIV. Very cool stuff. Favorite abiogenesis hypothesis. I'm weird. I don't know how familiar people are with this, but with abiogenesis, with how life emerged. I did a whole series on this about how that one is the only hypothesis. They're not theories yet. They're still not hypotheses.
Yeah, there's a bunch of competing hypotheses with varying levels of support. And right now, so for a long time, the controversy was protein world versus RNA world. And now what we're seeing is it's probably some combination of those two. And you can get what are called ribozymes, which are RNA enzymes, and then simple polypeptides interacting with each other that can do some pretty complex things. But then the other side of that is a question of kind of metabolism first versus structures first. So do you get like the chemical reactions and then they end up in cells? Or do you have like proto-cell structures and then the metabolism develops inside them? And I started on the proto-cell side of things, and I've been convinced that metabolism first is more likely correct. That you've got just big kind of metacycles of chemical reactions, and then they get contained within like lipid structures later.
Yeah, that would be even though that would be probably very boring, not exciting, but it's probably more interesting to actually have two real competing views on a debate instead of just the usual evolution creationist debate. Because
I would watch the heck out of a 2-hour debate between a proto-cell first person and a metabolism first person. I don't know who those people would be, but I would love to just watch them just hash it out for 2 hours. I would love that.
To both actually have an actual. Either one could be right viewpoint instead of just
right. There's legit evidence for both. It's not that the thing with abiogenesis is it's not that we don't have a plausible model. It's that we have more than one and we don't know which one is best. And that's a really cool situation to be in.
With the RNA first thing, I always wondered if maybe but since RNA and DNA both have three similar what was called three letters in their thing are the same, but only one letter is different. I always wondered maybe if DNA became when the one letter in RNA mutated to form the DNA letter.
Okay, I got to tell this story because this is my favorite thing. Like, literally, this is you accidentally, you had no idea, you could never have known this, but you just hit on my, legitimately my favorite thing in biology, which is, so the letters, the bases for DNA, A, T, C, G. For RNA, it's A, U, C, G. It's uracil instead of thymine. Why is that? That's weird. Why is that? Now, everybody agrees, there's no real controversy that RNA was before DNA. We'll agree on that. The reason we switched from uracil to thymine is because of cytosine, because cytosine sucks. Cytosine is way less stable than the other bases, and when cytosine mutates, it mutates in a very specific way, really fast. It does this thing called deamination, and when it does that, it turns into uracil. So if you have a genome that has cytosine and uracil in it, and you come upon a uracil, you don't know if it should be there or if it should be cytosine. Now, the solution to that problem, this is a great example of evolution being a tinkerer rather than an engineer. Because an engineer would look at that problem and go, okay, this is dumb. Let's get rid of cytosine and find something that works better, that's more stable so it doesn't constantly mutate into this other thing that we use. But that's not the solution we found. The solution we found was get rid of thymine, or get rid of uracil and replace it with thymine. Because then, anytime you see uracil in your genome, you can go, aha, that should be cytosine. And to this day, every cellular form of life on Earth has enzymes. All they do, all day long, 24 hours a day, is they search your genome for uracil. And when they find it, they cut it out, and they replace it with cytosine. And that's all they do. That's it. And the reason that is the case is because cytosine is stupid. There's no reason cytosine should be in our DNA. It's dumb. We should have gotten rid of cytosine and kept uracil. And here we are. Thanks, evolution.
Yeah, I guess that technique's the intelligent design model.
Yeah, cool. And it's not, so the argument, and it's not, I want to be clear, it's not a this is bad design argument. The argument of why that whole thing supports evolution is it says, based on what we observe right now, that explains it, right? Yeah. That is a mechanistic explanation for why the genome works the way it does, right? It's an affirmative case. It's not saying, oh, well, that's stupid bad design, therefore intelligent design is wrong. It's saying evolutionary processes explain this thing that otherwise doesn't make sense.
Yeah,
it's so cool.
My favorite one, before we go, my favorite one that I've heard is a giraffe one, that thing in the giraffe's neck, everyone's neck.
Oh yeah, the recurrent laryngeal nerve. Yeah, right. Because it loops in that one little, it loops around that artery. So in most things, it doesn't matter because it goes here, it loops and it's back and it doesn't matter. But in a giraffe, yeah, it loops all the way down. So it goes down the neck and then back up the neck. Yeah. By the way, I just put in the chat on YouTube, there is a paper on this cytosine and uracil thing. It's called Confounded Cytosine with an exclamation point. That's actually the name of the paper. And it's one of my favorite science things that's ever been written. I just love it.
Aaron, 1 second. I will make you a moderator so you can put a link in there if you need to.
Oh yeah, I'll just Google it real fast and put a link in there. It's such a fun, I mean, I'm weird, but I think it's a fun read. By the way, I saw somebody had asked about favorite or best books on evolution. I always recommend to people a book called Your Inner Fish by Neil Shubin.
I have that book on Audible.
It's wonderful. It's it's very accessible. It's not overly technical, and it really does a great job. Neil Shubin just does a great job of making these connections in terms of the showing how, you could look at how limbs develop in fruit flies, and that tells you how limbs develop in mice and humans by extension. And it's just like, why? Why does that work? The answer is evolution. Is why that works. It's a great book. This book, Your Inner Fish. I cannot recommend it enough.
Oh yeah. I watched the 3-part series on whatever channel it was, like on YouTube or PBS or whatever it was, Discovery.
I have not watched.
Inner reptile, inner monkey. I think the different.
Right. Yeah. It goes. Yeah. I love it. I mean, that's one of the fun things with like evolution phylogenetics. Is like, yeah, we're humans, we're apes, we're monkeys, we're mammals, we're
tetrapods.
Fish. We're tetrapods, we're fish, we're chordates, we're this, that, the other thing. It just goes all the way up.
Yeah,
I love it. Yeah.
If we had to go, I never knew. I knew some of the steps, but I never knew there were so many steps between fish and actual tetrapod or between mammal and amniote.
Oh yeah, right. There's the ancestral amniotes, and and you could just slice it finer and finer every time. You specialize and just zoom in.
I knew about synapses, thorapsids, and cyodots, but there's still so many between those things.
That's about where I'm at. Like I got those. I got reptiles, I got mammals, and like that's what I got. The specialists, the people that really know this stuff, man, there's so many levels in there. It's really cool. It's so cool.
So you want to advertise? You have a Wednesday show every Wednesday or so often. You want to advertise your show?
I have an irregular schedule. I used to be a lot more regular in my schedule. I only started my channel back in I don't even remember, May, June, sometime in there. I used to be a lot more regular. But then what happened was my wife and I had a baby, our second, in August, and then
kids can do that.
And then my family leave ended in October, so I went back to work. And I teach biology at the college level. So I do a lot of video recording and producing these days, but it's all for class instead of my YouTube channel. But I try to do stuff once a week or every other week. A lot of what I've been doing lately is more like this, kind of streaming and collaborating rather than like recording and editing stuff, because that just takes a lot more time. But my channel is Creation Myths. It's just Creation Myths. And it's all about basically taking creation science, young earth creationism, like at face value as science, and let's just examine it as science and show that like none of this makes any sense. It's just completely unconnected to like how things like population genetics actually work. Bears no relationship to reality. But we're going to approach it like we're actually going to do the math, and we're going to show that these hypotheses do not work. Like I know it's I'm sure this is great marketing, but like a significant number of my videos, like we do math, which like come watch my videos. There's math, right? It's really good marketing, but like that's what it is. We're really going to treat this as science and see how it holds up or doesn't. And the answer is it doesn't. So that's what I do. And
I tried to have schedules if I can help it, but like a few years ago I tried to have at least once a week, but that turned out to be a little, especially when I didn't have really regular guests, and sometimes I had to make up stuff on my own just to get a product out there. Like, so it burnt me out a little bit.
I know the feeling. Yep.
Yeah. Well, I'll let you go to bed because you have to keep.
I'm going to go to bed, everybody. It is my bedtime.
Yeah. So, before we go, do you have a catchphrase you say at the end of your videos to say goodbye?
I always tell people to don't get fooled. Don't get fooled.
Right.
That's it.
As I always say, never stop learning and enjoy the randomness. We'll see you next time. Bye.
Thanks, everybody.